The overall goal of this procedure is to generate concatemerized DNA that can be used to reproducibly stimulate intracellular innate immune DNA sensing pathways. This is accomplished by annealing the forward and reverse oligonucleotide primers and subsequent blunt end ligation to form concatemers. Then, the concatemerized DNA is mixed with cationic lipid reagent to form transfection complexes. The transfection particles are then added to cells for in vitro transfection or injected into the ear pinna of a mouse. Ultimately, quantitative RT-PCR is used to show the successful induction of an innate immune response by measuring the transcription of cytokines and chemokines.
This method can help answer key questions in innate immunity, such as how cells and organisms respond to self and foreign nucleic acids. Demonstrating this procedure will be Christian, a graduate student from my laboratory.
All animal studies used in this manuscript were performed under approved institutional protocols and animal care guidelines. To begin, resuspend the forward and reverse primers in molecular biology grade H2O to a concentration of 10 micrograms per microliter. Then, mix 5 microliters of the forward primer with 5 microliters of the reverse primer in a 1.5 milliliter microcentrifuge tube. Heat the primer mixture to the correct annealing temperature for 15 minutes. Then, place the mixture on the bench to cool down at room temperature. While the mixture is cooling, prepare 60% PEG 8000 by dissolving 3 grams of PEG 8000